So next we have a couple of related presentations. And this, you want to do it? All right, go ahead.
So we decided to use, again, the significance of this 100th meeting of this advisory council to make a presentation I've been wanting to have made for a couple of years, but I thought the timing was good now, even though I know it's getting late in the day. As you can tell from my director's report, as you can tell from a lot of the stylistic and cultural things that I emphasize in the Institute, I really value our communications and our outreach efforts writ large. I think it's really important. I think the field of genomics deserves it. I think NHGRI deserves it. And 4.5 years ago, I recruited Sarah Bates to take over what was then called it was actually a different name. We've subsequently renamed it to the Office of Communication. I recruited her from the National Science Foundation. She has a physics background. She does not know genetics and genomics. But the major charge that I gave her was I thought we had all the pieces, but I thought that the whole could be greater than the sum of the parts. And I think what Sarah has done under her leadership is to make the whole stronger, more robust than a simple summing of the parts. She's going to just, and I think she did inherit a number of key and novel pieces, including the history program, which this council has heard from, because not this council, a previous council, because I think you've all turned over. About 5 or 6 years ago, you heard a presentation from our Institute's historian, Christopher Donohue, who you'll hear from briefly. But I just thought it would be fun to have Sarah give a broad overview of the Office of Communications and what she has done to put it together in a different way, in an integrated way. Hear a little bit from Chris Donohue, but also we have a couple of guests here who are scholars who are working on some of the historical materials we have put together. And so I'm going to turn this over to Sarah. She's going to MC the next hour.
Thank you, Eric. It's nice to see everyone. You might know me from my time on top of the St. Louis Arch. And I was actually so much more nervous about the cake today than this presentation, so I feel really calm now. Just the cake operation going well. So I'm going to talk for the next 15 minutes, give you an overview of what NHGRI does communications-wise, big C communications, and then I'm going to turn over to my colleagues here. I understand we are short on time, but this will be worth it. So everything we do is in alignment with NHGRI strategic vision. And more than that, we have sort of guiding principles that we adhere to, guiding values, guiding principles for everything we do in communications. I have a fantastic job. I really appreciated what Betty said earlier about being in a place where you really believe in the mission and you really believe in your colleagues and you work together as a team. My office works incredibly well together as a team, and I think that reflects how the Institute overall operates, and a lot of how we operate in the Office of Communications really reflects how genomics as a field and NHGRI as an institute work together. So all of the things that you see up here should be pretty familiar. I think they reflect a lot of the presentations you've already seen today. Communications, thanks to Eric and the rest of the staff at NHGRI, is a really integral part of a lot of the programmatic activities that go on. We're brought in really on the ground floor of almost everything that happens, and I think that results in a much stronger outreach and operational product across the board. I'll talk a little bit, too, about how we are newly integrating more social, historical, and ethical context into all of our products, and then some of the tools and their changing uses across the field. And then I'm going to talk a little bit about collaboration, and I'm going to go into one case study in particular that you will find familiar, I hope. So as Eric said, about 5 years ago, I started with NHGRI. At that time, the history program was a part of the office, but it was kind of hanging out there a little bit. And since then, we've really focused on integrating history more into everything we do. And that sort of brought everything alive in a new way. And it's really set a really exciting new way of doing science communications because it bases everything in this really strong historical and social and ethical context. So I'm going to be giving this, broad overview of communications, and then my colleague Christopher Donohue is going to go into the history program, so you can learn about the program specifically. And then Spencer Hong and Thomas Schroeger are going to be going into even more detail about the really cool research that they're doing. So this is just to give you an idea of what the team looks like in the organizational structure. We have almost all the presentations you saw today pull from some of the materials that these really talented people put together. So multimedia, graphic design, science writing, web design, public affairs, media relations. Daryl Asia designed the top of the cake. No one baked the cake. That's Wegman's job. And we haven't, we did restructure a few years ago into this team-based system, but it's really an integrated office overall. There's really not any siloing. Everybody works together. The history team is kind of floating in between all of the other teams, but it's really an orchestra that plays together. I'm really just one of the conductors. This is what everyone looks like. We have a couple new people who you saw today. Theo Tiffany and Liz Dietz recently joined us as communication specialist and a new fellow. And the team that you saw there is really at the forefront of modern science communications. And the field as it is very different now than it was 5, 10, 30 years ago. It's not putting out one press release and watching media pick it up. It's really, you have to put out products across all the channels, and you have to make sure that you're really engaging with those different audiences on those channels. You have to make sure that you're aware of the social context that's happening and what's happening in the news. You have to follow the trends. And you have to make sure that you're reaching diverse audiences. You have to make sure that your content is accessible, that it's not just a certain type of person who reads genomic news. It's really a lot of new audiences that we're trying to build into this genomics community. And we do this through a wide variety of platforms. So I'm just gonna give you a little bit of a sample menu, a little taste here. We have traditional press releases, of course, which I'm sure you all read weekly. We have the NHGRI brochure, which we recently redid. I'm sure that's all framed on your walls, on your offices. We recently redid the Talking Glossary of Genomic and Genetic Terms, which is one of our most popular features on genome.gov. We are present on LinkedIn, Twitter, Facebook, and Instagram. And so we post every day on all of those platforms, and we tailor the content to each platform. So each of these platforms do have different audiences, different ages, different interests, and we really alter our content to really fit those different platforms. We have a lot of basic explainers. The population descriptors explainer was just mentioned. We also do a lot of fact sheets. Genomics and diversity in the workplace is one that's been popular. We are also on Flickr because we have such a talented group that does a lot of visual design. So illustration, animation, these are all on our Flickr page. If you're ever interested in grabbing something, it's all there for public use, Creative Commons. Just please credit us. And we're investing particularly this year in our YouTube channel. So YouTube is increasingly a social media platform. People kind of forget it's a social media platform, but it really is supposed to be an engaging platform where you're looking at the comments. We do look at the comments, for better or for worse, and we try to respond to them as much as we can. And we're focusing more on these shorter, like bite-sized videos. You can see our science writer there, Anna Rogers, who just put out a short piece on why the Y chromosome took so long to sequence. And of course, we have our main hub, genome.gov, which, as Eric said in his director's report, was recently refreshed. We already have a 30% increase in traffic from Google just because of that refresh, so our development team is doing a great job in increasing our search engine optimization. We're not only competing successfully, and we're not only competing in the science communication landscape, we're really competing successfully in the landscape. So we've been recognized outside of NIH and HGRI by entities that recognize government agencies that do creative work in science communications. Of course, we've also been recognized inside NIH, particularly for the History of Eugenics and Scientific Racism Symposium that was led by the history team in 2022. So the case study I'm going to talk about is, can anyone guess? Pangenome. Yes, thank you. Yes, you get an extra point there. So pangenome, every year we try to identify 2 or 3 big findings or discoveries or exciting genomic initiatives that we can really focus on and put all of our energy behind communications-wise to break through the noise even more online. So pangenome, of course, was a fantastic opportunity to do it. We followed it ever since it was approved by council as a concept, the Human Genome Reference Program. We knew that this was coming, so starting years ago, we started putting out, material that was starting at the beginning. Lots of people don't even know what a human genome is. So for some of our audiences, we have a fact sheet on what is genomic variation that explains, okay, what is a human genome? What is a reference None of the Lyft drivers I ask know what a reference is. And then why do we even need a new human pangenome reference? So these are all very basic informational products that we can put out without breaking any embargoes, most importantly on any papers that are coming out, and it really gives us an opportunity to set like an educational stage. We also focus a lot, as I said, on multimedia. So the pangenome video we put out, almost 2 years ago, and then only two or 3 months ago, we put out the actual pangenome announcement itself. So this was one of our most popular videos. This was what was recognized by the DC Science Writers Association. And then it got 10,000 more views the day we actually announced the new pangenome reference. We have a lot of fun in communications where we explain to people it's actually really simple to sequence a human genome. It only takes 7 easy steps. Oh, wow, you really do think it's easy, huh? So Adam Philippi was really a good sport in agreeing to be in this video with us. If you haven't seen the video, I really encourage you to watch it. At the end, he gives like a really nice wink that we somehow coerced him into doing. We also are constantly putting out information across our different platforms. Does anybody on council follow us on social media? Oh, Sharon Plon rolled out, that's why. Oh, there we go, thank you. Okay, by the end of this talk, I'm sure everybody else will too. So we are on Twitter. We're kind of moving away from Twitter, more onto some other platforms. But at the time, Twitter was still going pretty strong back in March. So we had a lot of posts that were focused more on sort of more somber, authoritative posts that were focused on the pangenome. And then we had very zeitgeisty social media meme type of posts. And this is something that has shown to be very popular, especially with more early career people. We also learn a lot from our campaigns. So in looking at the engagement on social media and in the press, we see where people are confused about certain concepts, and then we go back and we try to iterate on how we can explain those concepts better. So this first tweet that you see over there on your left is actually an illustration that we did right after the pangenome announcement, because we found people were confused about what diversity really means. What does diversity mean? When you're talking about diversity in context of the pangenome, you're kind of talking about 2 or 3 different things that people think that they're familiar with, and we are trying to disambiguate that a little bit. Of course, outreach when it comes to press coverage has the most variables because you don't know how press is going to cover a story. We can put out something in a press release or on social media and we can fact check that 1,000 times with our scientists, but once you hand everything over to the press, they're going to cover it however they like. So you can see here there's sort of a wide variety of the ways that the press decided to cover the pangenome. Overall, we were very happy with it because it actually talks about diversity. It talks about, the need to do this to improve human health, or it's connected to genomic medicine. So these are all main messages that we're also looking for, in addition to lots of clips that come out. So we have a lot of fun, but we do measure our impact in a very meticulous way because we want to establish baselines for ourselves so we can continue to improve. And then we also just want to figure out where people are talking about things, where our target audience is. So we use a variety of monitoring tools. I won't go into all of them here, but it really is focused on looking at the website, social media, and news engagement. So you don't have to look too far into the different axes on this, but what you should pay attention to, or what the main takeaway is that this is just the results for looking for the word pangenome in news clips over this amount of time. So this is just over, just a few days. We put out the pangenome release early in the month, and then you see it really spiked up to a reach of about 1.2 million. And these are numbers that come from the news circulation industry. So they're a little bit inflated, so don't pay too much attention to the billions and billions of people that BBC says that you're reaching, because some of that's for advertisers. But what we do is look at like relative peaks, right, to see how one campaign is doing compared to another. We also look at what outlets are covering us and what their circulation numbers are too, and the types of coverage that they do. So when we do a lot of our proactive outreach and we do a lot of relationship building with different members of the press, that we can build targeted relationships more based on the coverage that they have. And then of course we want to know how things like pangenome compare to the first full human genome sequence that was announced by the Telomere to Telomere Consortium. So what we do is we really get into it and look at how the peaks vary. You know, the peaks correlate with things like, are we getting one news bump? Are we getting two news bumps? Are they reporting on the finding initially and then doing a deeper dive into what does it actually mean in a social level? So these are all different types of stories that we really work on very strategically with members of the media. I just wanted to share this. It's one of my favorite memes that our social media person put out. I also just wanted to mention the Y chromosome campaign because that was so recent. So we messaged the Y chromosome. It's the first full sequence of the Y chromosome. We spent a lot of time ahead of time talking about how we were going to talk about this because it's such a fraught topic right now in the news. Issues surrounding gender and sexuality are on the minds of policymakers, are on the minds of voters. And so we knew that this might be misconstrued if we just put it out there into the world. So we spent a lot of time talking to our colleagues in the NIH Sexual and Gender Minority Health Research Office to review language, talking to our colleagues at Building 1 and NIH, the mothership. And we go out of our way to make sure that we include extra language in here, you know, that what is social category, what is not. And now I'm going to turn it over to my colleague, Christopher Donohue, because this is a good place to show why history and social and ethical context are important.
Okay, all right. Thanks so much, Sarah. All right, so I'm Christopher Donohue. I'm a historian of genetics and genomics. I also focus on the history of eugenics and scientific racism. And I received my PhD from the University of Maryland, and I co-lead the History of Genomics program with Brittany Kish. So just as a little bit of background, Eric founded the history program in 2012 to save and later make available the critical materials outlining the history of the NIH role in the Human Genome Project and its ongoing guidance of genomics. So on the left, you actually see the very first efforts at capturing the history of genomics by Francis Collins himself. So that's a CD on the left. But Eric Green understood that we really needed to do more, and he hired me, and working with Chris Wetterstrand, many of the people in this room, I've been working here as the historian ever since. So on the right, in the center, there's the picture of Zach Utz, who is our current archivist, and he is our public historian. And there are still remaining archival collections to be digitized. Our full archive, I should mention, contains over 1 million pages of materials from the beginning of the Human Genome Project to the present. And we are one of the largest digital archives in biology and genetics, and the only one embedded in an NIH institute. And we are probably the only exclusive genomics archive in the world. And as importantly, because we are almost exclusively a digital archive, this is the picture on the right, that's actually not our server room, obviously. Scholars don't require grants or travel to do research, but we do occasionally welcome them to campus. All of these features together are meant to illustrate that we are universally recognized in the scholarly community for many of our cutting-edge approaches. And in fact, our collaboration with the Amaral Lab, which you will hear about shortly, is emblematic of our ability to leverage the unique resources of the NHGRI and the unique resources of the Office of Communications. So while we are continually archiving, we also realized that we needed to capture the experiences of retiring staff and key members of the genomics community in real time. So around 2015, we established an oral history program. So we have an extensive oral history program that you actually see a clip of the YouTube video with Eric Jarvis. And we have more than 50 oral histories published, with close to 100 being planned or recorded, with many more being readied for publication on our Genome TV YouTube. We also have had a very well-attended series of public-facing events on Zoom. So you see, for example, our roundtable discussion on the promises and perils of social and behavioral genomics that we held earlier this year, and we also have had a number of interventions on complex topics like the complex connections between the history of eugenics and scientific racism and genomics. And often we have more than 1,000 people actually in the audiences of these events. We also have developed fact sheets, and this, like our public-facing events, illustrate the unique synergies between history and communications at the, as part of the Office of Communications, as part of the NHGRI. So as I discussed in the previous slide, we have an amazing archive of unique materials. And as a result, over the last decade, we have had a very active network of researchers using our archival materials to publish peer-reviewed books, articles, other publications, both popular and academic. So the two examples here are James Terryberry's Tyranny of the Gene and Sonia de Chaudhavarian's Heredity Under the Microscope, which was published in 2021. So since we have such a unique archival resource, it's important that we not only have robust ethics as our basis, not only for our collections, but for our research community and any analysis and use of our files. So both the Archive Space project that you heard earlier about in council and the machine learning collaboration with the Amaral Lab are guided by FAIR principles, maximizing openness, transparency, and accountability. Soon, we want to make more essential files available to the public through Archive Space, while also using our insights from the Amaral Lab to enrich our history and further develop our scholarly collaborations. And as a program, we are committed to continuing to ask and answering the big complex questions about the history of the Human Genome Project and genomics, as well as how team science works in biology and genetics. And in order to achieve this, as you heard about a little bit earlier, we've developed actually a dual space for our archival documents, one of which is archive space, the other of which is a more controlled access space due to the very nature of government documents. So through archive space, the image on the left, we're making a small number of our most important files, like those from past leadership, like Francis Collins and Ilka Jordan, publicly accessible. The second is a core collection, which is accessible through SharePoint, through an institutional collaboration agreement of our network and community of researchers, which has about 5,000 files, which is the basis for the Amaral Lab analysis that Spencer Thomas will talk about very shortly. Thus, it is my distinct honor to introduce Spencer and Hong and Thomas Steuger of Northwestern University, who will now discuss the presentation on the right, which comes out of the core collection of our files available to the research community through SharePoint. Thank you very much.